Good afternoon. Today we will be teaching you about meiosis.
Alright, so talking about meiosis. So, meiosis is basically when the DNA is replicated, so basically it splits apart into two other new cells. And basically, the chromosomes that are in this cell, they'll just end up pairing with other random genetic combinations. So it's like a mix and match. So going into it, we have interphase, which is when the cell is just like this. It's not really doing anything. It's just kind of laying about. But now we transition into when an egg is fertilized by a sperm, which is now prophase. So now in prophase, we basically have the DNA. It's just going to be replicated. Like the DNA has already been replicated during interphase. So now the process is now we have 4 of our DNAs right here. One is from your dad, one is hopefully from your mom, and now we have to make spindles and other tetrads, which will help break up the chromatids and pair them up randomly. That'll happen later though, but right now we're just getting everything set up. So during this process, it's called crossing over, which basically means that, let me see what crossing over means. It's basically just when just a genetic recombination. It's just swapping chromatids between each other. So now going into metaphase 1, there are 2 phases to meiosis, which is kind of interesting. Metaphase is basically when the nuclear envelope that prevents these from mixing together, they disappear. So now these DNA strands, 2 of them are from your mom, 2 are from your dad. They will pair up right here, and those little things that are fusing together, these spindles will end up attaching onto the center line of your chromatids. This plate right here, this is where they pair up. This is where they get in line. It's called the metaphase plate. So in the center is the metaphase plate. There we go. The connector cores, which are these little thingies, they attach to the ends right here. And they're going to pull them out. They're basically going to pull them and make different strands of DNA with different combinations. And these, just want to say, these tetrads, they randomly line up with the metaphase plate and they attach to the spindle. So now anaphase is when we actually start splitting the cells, or splitting the DNA strands. So these are called chiasmata or something. And they split them apart. The spindles, the tetrads, they just pull apart the DNA and they just kind of retreat over here into their new other chromosomes. So now they're haploid cells, which now they have 46 chromosomes. No, I'm right. Never mind. I'm right. Telophase is now, these cells will now create a cleavage furrow. This cleavage furrow will help separate the 2 cells, because now the 2 cells have their own DNA pairs. So now they got their own daughter pairs, which are different chromosomes that have been mixed during metaphase and anaphase. So now this cleavage furrow is made in order to split the cells apart. And yeah. Alright, so now into meiosis 2, we're after interphase, we get into prophase 2. So prophase 2 basically there's no real cell, chromosome duplication. We have the 4 chromosomes are right here. We have 2 diploid cells, my bad. And these centrals duplicate by separating the members and going to opposite sides. So these will end up going over here, and these will end up going over here. And then the nuclear envelope just breaks down, allowing these 2 to just go to opposite poles. Alright, and now we go on to metaphase, when each daughter cell completes forming the spindle apparatus. So the spindle right here, it ends up linking on to the chromosomes right here. And these chromosomes will wind up at the metaphase plate. So basically the center of the cell. And the connected cores, which end up lining up right here, will face the opposite poles to be pulled during anaphase. Anaphase is when the centromeres separate and then the chromatids move to opposite sides of the cell. So if you were pulling if you're pulling the X apart, you would just pull both of these cells, both of the chromosomes out. And then telophase is when the nuclear envelope forms back, keeping the two haploid chromosomes together, and then it creates a cleavage furrow to then break up the cells, creating 4 healthy haploid cells. And that's what happens in cytokinesis, where the end product should just be 4 haploid cells, each cell containing 23 chromosomes, and that is the end of meiosis.